Hello, shall we look at the pancreas under the microscope? Some more classic histology. So the pancreas is an organ of the gastrointestinal tract, and it has two jobs. It has exocrine jobs and endocrine jobs, which we'll talk a little bit about. When the pancreas in humans formed in the embryo, you had two pancreases, and as everything rotated they came together, which is why we see a main pancreatic duct and an accessory pancreatic duct. But the reason it's such classic histology is because when we look down the microscope we'll see that it is almost entirely, it's glandular. The pancreas is a gland. It looks very glandular. Most of it is the exocrine gland secreting enzymes into the duodenum, into the small intestine, that are going to help with digestion. And then there are little islets, tiny little islands scattered around of the cells that do the endocrine bit that manage blood sugar. Okay, what do we see? See a big purple blob. Okay, that's for later. There's one on the lowest power here 4 times, and it looks very homogeneous. It looks very similar with some streaks of lighter colour running through it. I think you can see what I mean by the islets. We also have some tubes in here as well, some big tubes and some little tubes. We need to work out what that all is. We've got a bit of a fold in there, which is a sectioning or a staining artefact. That'll be a sectioning artefact. So this is what we mean by something looking glandy, something looking like a gland. We've got an awful lot of cells in here, and the cells in here, most of them, so we're seeing like pink circles with a purple ring around the outside. Those are the exocrine cells. Those are the cells that are going to be making digestive enzymes and passing them outside to the pancreas. So this is what we mean by glandy. All these cells in here are making things. It looks very, very glandy. And they're in circles because those cells are arranged around a duct. So that what they secrete goes into that duct, then out through other ducts, and eventually into the duodenum. So that's an interesting idea. We looked at the histology of the gastrointestinal tract, and we talked about how it's a tube within us. It's an external surface within us, if you think about it, right? It's continuous all the way through. Now, if you think about that gastrointestinal tract, and you think about these accessory organs that are going to secrete into that tube, well, these ducts in here are also external tubes, external surfaces inside us, because they are continuous with that gastrointestinal tract. These are continuous with the pancreatic duct that is ducting into the duodenum. Quite the concept. So yeah, what we're seeing here is we're seeing more epithelia, and epithelia that are, like I say, secreting various enzymes, which is a whole other problem for the pancreas. But let's zoom in again. So this is now my 20 times objective. So this is 200 times magnification to my eyes. How much it's magnified for you will depend upon the size of your screen. But I think we're getting the general impression here of lots of circles. I'm trying to find some just very neat ones. They're all kind of neat. There's a good one in the middle. There's a few there in the middle. Let's jump up to what's this? 40 times objective. Don't know how clear this is going to be. This is an acinus. These are acinar cells. An acinus is anything that looks like kind of a berry. So like cells clump together to look like a berry is an acinus. So these are acinar cells forming the acinus, and really they are kind of pyramid shaped, and their fat end, their base is away from the lumen, and their narrow end is at the lumen. All right, can you see, in fact there's a couple there, maybe, but that one in the middle there is the best one. Can you see there's a white center, so that's the duct. And then around it we have a circle of cells. We can see that the nucleus is at the base of the cell, so that's the flat base of the cell. These cells are pyramid shaped. We're only looking at them in 2 dimensions because we've cut a section through them. Can you imagine them in 3 dimensions, kind of making a ball there? So the base of the cell is purple, and then we can see the rest of the cell is very, very pink. And the pink part is the apex of the cell, which is going up towards that duct, towards the lumen there. It's pink because the cell there is filled with granules, zymogen granules. We've got inactivated enzymes in there, which are packaged up and ready to go and will be released when they're instructed to be released, and then they can go into the duodenum and help with digestion. The duct there is called the intercalated duct, and these ducts, they're all joined up, these intercalated ducts, and they will eventually lead to larger ducts, which are sensibly named. So you have intralobular ducts within a lobule, interlobular ducts between the lobules, which then go to the main pancreatic duct, or the accessory pancreatic duct, which then run to the duodenum, ampulla ovata, sphincter of Oddi, open into the duodenum. We'll see if we can find examples of those later. But this is what we mean by exocrine pancreas. These coloured cells here, these circles of cells, these spheres of cells really, these balls of cells. And look how most of the pancreas is arranged like this. And most of the pancreas's job is making exocrine secretions. That means we're going to secrete these enzymes onto an external surface. The gastrointestinal tract is considered that external surface inside us. So these exocrine secretions will then digest, break up proteins, fats, carbohydrates, DNA and RNA even. And as I scroll around, we see these lighter patches, don't we, which I'll come back to later. Those are the endocrine parts. Okay, so we're going to see some good blood vessels, and we're going to see some good ducts. Maybe I should zoom out a little bit and drop that brightness. So the pancreas is well vascularized, so we're seeing lots of blood vessels. I'm going with okay, so here we've got, we can see an endothelium, and we can see smooth muscle, and we can see a thin-walled vessel with a thicker-walled vessel. So we're going with a pair of blood vessels there, an arteriole and a venule. Whereas if we roam around, yeah, look, can you see how that's different, right? That doesn't look like a blood vessel. So remember, these ducts are all lined by epithelium because they're a continuation of the gastrointestinal tract. So the intercalated ducts, we might see a simple cuboidal epithelium, and then as we get to the interlobular ducts, we find that epithelium gets a little bit taller. So a simple but quite low columnar epithelium, so a single layer of columnar epithelial cells, and they're not stained like the acinar cells, the secretory cells, are they? They look a bit different there. So that's what the ducts look like. But really, I think it's the exocrine cells that are most interesting, right? And now you're starting to see, maybe you can see the difference between blood vessels and ducts. So the other thing about the duct is, of course, there isn't very much connective tissue around it there. But as we get to the bigger ducts, we saw a huge one earlier, didn't we? Where was it? Yeah, look at that. That is, if you remember the blood vessels video, that is just so not a blood vessel. It's a tube, it's made up of cells, but how do we know that's not a blood vessel? When we look at blood vessels, we see that they've got thin, flat endothelial cells on them. But as we zoom into this, because we've been looking at epithelium so much over the last few weeks, we've been looking at the GI tract, we see the same stuff here, don't we? Look at that. That is clearly an epithelium. That's not an endothelium, that's not a blood vessel. Those are columnar cells, so those are tall cells, so it's a columnar epithelium, and it doesn't look quite simple anymore. It looks like we've got kind of probably more than one layer of cells there. So this is a stratified epithelium, a stratified columnar epithelium. So this clearly is one of the main ducts, one of the big ducts. We can see little ducts all around it as well, right? Little ducts running with it, which will eventually drain into it. That's what we're seeing around here with these other circles. So we're seeing one of the pancreatic ducts, because like I say, this is the epithelium, continues with the epithelium of the GI tract, which is why we see this epithelium, which looks like the epithelium of the GI tract inside the ducts of the pancreas. So that's how we know what it is. And as we slide outwards, there's a lot of connective tissue here, a lot of collagen supporting this. So to be honest, this looks like a main pancreatic duct because of that stratified nature of the columnar epithelial cells and all of that collagen. If we go back to the lowest power, how is it size-wise? Well, I don't know what animal this is from, but it's still pretty diddy. But so maybe an interlobular duct, but it's a pretty good one. And then if we slide up the top there, look, we can see another tube that's kind of in between the small tubes and the big tubes, and we can see some similar features here. That's very pretty. So again, we can see that the inside of that tube is lined by an epithelium, and that epithelium is looking a bit columnar, and we're seeing layers and layers of connective tissue around the outside. So maybe an interlobular duct. So we go from the intercalated ducts, which are within all those acinuses, to interlobular ducts, which are the slightly bigger ones. Sorry, intralobular ducts, which are slightly bigger, and then the interlobular ducts, and then the main pancreatic duct. That's the method. So what are these cells secreting? Not these cells, but these cells. Well, the pancreas has a problem. The pancreas would very much like to make enzymes that digest proteins and carbohydrates and fats, and we are made of proteins and some fats, and we like to store carbohydrates. So these cells in here, well, do you remember when we looked at the duodenum, we saw the enteroendocrine cells? So endocrine cells in the intestinal crypts, and those enteroendocrine cells would send signals to control parts of the digestive process and absorption process. Well, cholecystokinin, CCK, is produced by those cells and tells these cells to release their trypsinogen, their chymotrypsinogen, their procarboxypeptidase, their proaminopeptidase, their lipases, their alpha amylase, ribonuclease, deoxyribonuclease, a whole host of enzymes that will be useful in digesting proteins to amino acids and carbohydrates down to sugars and lipids, fats down to lipids. But the problem is, well, what's happening here is that the enzymes that get released from these cells will be inactive precursors of those enzymes. They will be packaged up in a safe manner, and then they will pass down through the duct system and out into the duodenum. And when they're in the duodenum, then they will become activated. And when they're activated, then they will start digesting proteins and fats and carbohydrates. So that means they won't digest the proteins here in the pancreas. The pancreas should be safe. They should only be activated when they enter the duodenum. The secretion here is also a little bit alkaline because the stomach contents were a little bit acidic, so that alkaline is going to change the pH of the contents of the duodenum. So the way you activate these enzymes is enzymes work largely. It's the shape of the enzyme. The enzyme is a protein. The shape of the protein attaches to another protein. That's how it works. So to activate the enzyme, you chop bits off, and it reshapes itself, and then it works. It functions. So that's the exocrine part of the pancreas. Most of the pancreas is exocrine pancreas, is involved in digestion, producing enzymes to help with digestion, which then get absorbed in the small intestine. What about these islets of Langerhans then? So we're seeing them scattered about all over the place. There's a nice one, there's a little one. Again, you've got to imagine these in 3D, so sometimes we're just kind of getting a little bit of it, sometimes we're seeing a lot of it. But these islets of Langerhans, look, there's a duct there, blood vessel, sorry. But these islets of Langerhans are much paler staining, and they are spattered about inside the pancreas. So all these cells have come from the same embryological origin. They've just taken different differentiation pathways. So these endocrine cells, that means that these are cells that are producing hormones. They are producing proteins that go into the blood and pass through the cardiovascular system to affect cells at a distance. So these are the cells that are making insulin and glucagon and somatostatin and pancreatic polypeptide. So there are different types of cells in here. There are 4 types of cells that we usually talk about. I am not going to be able to identify them. I think the only way I would confidently be able to tell them apart is if I used immunohistochemistry or in situ hybridization to see what genes or proteins they were producing. They're not very well staining, and they're all, blobbed together there. But we have the alpha cells, beta cells, delta cells, and PP cells. So alpha cells will produce glucagon. Glucagon will tell cells around the body, so it'll encourage the liver to release glucose from stores, from glycogen stores, and other tissues like muscle to release glucose. So alpha cells release glucagon, which tells tissues of the body to put glucose into the blood to raise blood glucose. Beta cells produce insulin. Insulin has a lot of jobs. Was it the first? Insulin was discovered really early. It's one of those really early protein stories. But insulin will encourage the liver, muscle, fat cells around the body to take glucose out of the blood and store it. Delta cells produce somatostatin. Somatostatin is produced by other tissues as well, but somatostatin will turn off digestion. So you've eaten your meal, you've digested most of it. Somatostatin is then going to start switching everything off, slowing everything down. And pancreatic polypeptide, made by PP cells or pancreatic polypeptide cells, will do similar. So pancreatic polypeptide will turn down stomach motility and gallbladder activity, and again switching things off after digestion. Oh, it's also going to switch off the exocrine pancreatic cells from doing their secretions. Somatostatin will also turn down insulin and glucagon secretions. So it's these guys, it's the cells here that are responsible for diabetes then, or certainly type 1 diabetes. So in type 1 diabetes, it's a bit of an autoimmune disease. So the beta cells here that produce insulin are destroyed. So there aren't enough cells. So you can't make enough insulin to get the liver and other tissues of the body to take the sugar out of the blood and put it into storage. That's type 1 diabetes. Type 2 diabetes is similar, but some of the cells here die, but some of the beta cells here die, the insulin producing cells. But there are still beta cells here producing insulin, but they're not able to produce enough insulin to do the job properly. And this seems to be linked with an insensitivity to insulin by the other cells of the body that should be taking sugar out of the blood and storing it. But like I say, the insulin does more. This is kind of the brief story. So type 2 diabetes, some of the beta cells are lost. The beta cells that remain don't produce as much insulin as they should, and so the sensitivity bit means that these cells are producing insulin, it goes off around the body in the blood, and the other cells then have a receptor for that insulin, and when they receive the insulin, they do a job, they become less sensitive to that insulin as a signal. So that's a good point actually. The other thing we can't see here, maybe we can get a hint at, because I can see, I think that's a capillary over there to the right, but the islets of Langerhans have tiny little capillaries in them, tiny little blood vessels. We would call these fenestrated capillaries. They've got little tiny holes in them. Fenestrated capillaries, what are the holes? They're like a couple of nanometers or something, but tiny little holes. So fenestrated means windows. So the capillaries in the islets of Langerhans take those hormones into the blood and then pass them off around the body. So fenestrated capillaries in the islets of Langerhans. But that's it. That is the microscopic anatomy of the pancreas, which no doubt I've over-egged again, but it's a nice looking tissue, isn't it? So most of the tissue, most of the pancreas, it looks very glandy. Most of it is that exocrine pancreas, that red and purple, with those very pale staling islets of Langerhans doing the endocrine bit, dotted around like little islands within an exocrine sea. And then this is all broken up by blood vessels and ducts carrying away those exocrine secretions to the duodenum so they can do their job in the gastrointestinal tract. All right. Again, we talk about insulin, we talk about diabetes, but it's the cells, it's the microscopic anatomy that explains all this. So I hope that was useful. I hope it was interesting. It's a nice tissue to look at. Okay, see you next week.